So again, a warm welcome to today's iCMLf conversation, featuring key biological and clinical highlights from the recent John Goldman conference on CML. And this webinar is for those of you who were not able to attend, or for those of you who'd like to get a recap or having a discussion on the sessions and the highlights with our experts today. We're really, really privileged to have, again, two co-chairs of meeting with us here today. So please welcome Professor Jorge Cortes from Augusta in the United States and Professor Daniela Krause from Mainz in Germany. So how do we run the session? We want to start with a 15 minutes clinical overview from Professor Cortes, then move on directly to a biological overview from Professor Krause to bring these perspectives together. And then we have room for 15 minutes for a broader discussion with all of you. So now it's my pleasure to introduce our first speaker. Professor Jorge Cortes will provide a clinical overview of the meeting. He is the director of the Georgia Cancer Center in Augusta, and before joining to the Georgia Cancer Center in 2019, he has been spending 23 years at the University of Texas in Houston, most recently as professor of medicine and deputy chair at the Department of Leukemia, where he directed the CML and AML program. As you may all know, he is an international leader in clinical research of CML, and he led many of the most important CML trials over the past decades. And he has authored or co-authored nearly 1,000 peer-reviewed articles during his whole career. As you may be heard of, he is also our new chairman of the foundation, and he's been co-chairing the annual John Goldman conference since 2014. We're really happy to have you here, and we're looking forward to your clinical overview now.
Thank you very much, Stephanie, and thank you everybody for joining and for spending a few minutes with us today. The John Goldman meeting this year was really outstanding. We had a great program. We had some fabulous presentations, a lot of new data, and this is very typical in this meeting. A lot of good discussion and a lot of new ideas and opportunities to collaborate and to develop new projects. So I want to highlight a few of the clinical studies that were presented during this meeting, and in some areas which I think are particularly of interest today. In the first session, in the session where we present the highest ranked abstracts, there were 3 studies that were presented that relate to frontline therapy, and this is important because, as you know, although we do have good frontline therapy in CML, there are still some gaps where we would like to improve when that is possible. One of them is that only a fraction of patients, probably at best half of the patients, get to a sustained deep molecular remission in the long term. And if we are aiming for treatment-free remission, we want to improve on these rates. So to that effect, one of the abstracts that was presented was the final results of a trial from Germany that's called the Tiger trial. And this trial, what our colleagues from Germany, and this was presented by Andreas Hockhaus, what they did is they randomized patients to receive Nilotinib alone or in combination with interferon. And we had seen early data from this study, but this is, again, their final analysis. And what they showed was that there was a significant, there was some earlier responses in the patients that received the interferon, but the absolute rate was really not that much different between the two arms. And in the end, they reached about the same level of the deep molecular responses and major molecular responses and so on. Now, what is interesting is that among the patients who discontinued therapy, the patients who received the combination with interferon, they seem to have a somewhat better probability of maintaining the remission after treatment discontinuation. So this is interesting, and there's been a lot of interest in the role of interferon in this context, and I find this very interesting and very stimulating. I think in practical terms, it's complicated to think that we would give interferon in combination with, let's say, in this case, nilotinib to every patient, but it may make us think about the possible role of interferon to try to improve the probability of maintaining remission after treatment discontinuation. There were two other studies also in the frontline setting during that session that were very interesting. One of them is, both of these studies are in using Asciminib. There's a lot of interest to use Asciminib as frontline therapy. We've seen the data of Asciminib as a very effective therapy for patients that have received multiple prior therapies or in patients that have a T315I mutation. It is established there. It's approved in many parts of the world and little by little getting approved in others, in other countries. But because of that high efficacy, because of the very good safety profile, there is interest in using it in the frontline setting. So the first study that I want to discuss is a study that's called the FASCINATION study. This is a study again from Germany, also presented by Andreas Hockhaus, and in this study they used a combination of Asciminib with nilotinib. And the rationale for this combination is that, as you recall, the binding site for Asciminib is different than the binding site for all of the other ATP competitive inhibitors. So there's in vitro data to suggest that there is synergy, that the combination would prevent the emergence of resistance and so on. So it was an important question to ask. And the results, unfortunately, show that there is too much toxicity, particularly myelosuppression, and that led to many patients having to stop therapy, to discontinue, and so on. So the results ended up being, this is a single-arm study, so all we can do is compare to historical data, but the results didn't seem to be, for example, the major molecular response at 1 year didn't seem to be that much higher than what we would expect with single agent, for example, nilotinib or another second-generation TKI. So, obviously the results were not what we would have hoped, but this was an important study to conduct because of that preclinical data that suggested that synergy. So, again, I don't think we would be using a combination frontline, but I think it was a very, very important study to conduct, and these studies then inform us for future studies. The other study comes from Australia. This is also a single-arm study, and in this study, the patients are treated all with Asciminib, and just like the Australian group has done in the past, the plan is to assess the responses at the given timelines that we know where we expect to give responses according to ELN and other recommendations. If there is no good response, then they would escalate the dose, and then ultimately they could even add nilotinib. But really, the majority of patients here are being treated with just single agent, and the starting dose initially was 40 milligrams twice daily, then was 80 milligrams once daily. The results are actually very impressive in this case. By 3 months, the early molecular response rate is in excess of 80%. It's almost 90%. And even at 3 months, almost half of the patients have a major molecular remission. So this is early data still, but it is suggestive that we are getting results that are better than we would expect with a second generation TKI or even with imatinib. And there are many other studies that are being conducted all over the world, including a randomized study comparing nilotinib, Asciminib to imatinib or to a second generation TKIs that will determine the true value of Asciminib in frontline. So very, very important studies, very valuable information. Another area that was discussed quite a bit during the meeting is the issue of molecular abnormalities, the mutations in other genes, in other cancer-related genes other than BCR-ABL. As we know, a number of patients have these abnormalities, and we've seen that these abnormalities are associated with a worse outcome, with worse probability of response, higher probability of progression. But there were two studies that I want to highlight here that relate to the significance in the context of treatment-free remission. One of them was presented by Sue Branford, and she assessed patients that had that were eligible for treatment-free remission and assessed whether they had clonal hematopoiesis. You know that this clonal hematopoiesis is present in many individuals. And it has implications because of the risk of developing cardiovascular complications and other hematologic malignancies and so on. So, of course, some patients with CML may have these clonal hematopoiesis. The interesting thing in this report was that those patients that had clonal hematopoiesis at the time of treatment-free remission, at the time they were attempting treatment discontinuation, that did not negatively impact the probability of remaining treatment-free. And actually, it was the opposite. These patients seem to have a better probability of remaining treatment-free. The mechanism why this happens, it's hard to understand. Of course, we need more data. We need to further explore these phenomenon. And the other thing that's interesting is then to see what happens in the long term, because of course it may improve the probability of treatment-free remission, but are these patients in the long term are going to have other hematologic malignancies, cardiovascular problems, and so on? We don't know that. Of course, the follow-up will tell us. There was another study, this one by Dennis Kim from Toronto, who also assessed something similar. In this case, whether the presence of these molecular mutations in patients that had that were attempting a treatment-free remission would have a negative impact. Now, this is the mutations, for example, in the ASXL and so on. And what he found is that, again, the presence of these mutations did not negatively affect the treatment-free remission. So it's interesting because those patients may have a lower probability of having a good response, but when they become eligible for treatment-free remission, they seem to have an equal probability of remaining treatment-free. And that also tied with a very good debate that we had between Jane Apperly and Beppe Saglio, where the question is then, should we be doing these next generation sequencing to all our patients with CML, considering that what we know about the importance of these mutations in other genes? Beppe emphasized the importance that these mutation, that this information has, the important prognostic implications that we already know that these abnormalities have, which make it an important piece of information for patients. Jane, however, argued that although we know that these have significant implications, there's really not a good understanding of what we should do about these patients, and that in the absence of real strategies that have been prospectively evaluated, it may be a little too early. And she emphasized that we're not ready yet to incorporate that into the clinic. So I think that, you know, it just this debate was a very nicely put way by both of them to emphasize that this is an area of active research where we definitely need to continue collecting information and learning more about, but so that we can get that information. And that, by the way, emphasizes the importance of one of our projects in the International CML Foundation, this Harmony collaboration, where we're trying to collect patients from all over the world with these molecular characterization, so that we can better address these questions, because only large amounts of data are going to be able to help us answer these questions. A couple more studies that I want to emphasize. There was a lot on TFR, so one of them was the very long, the final analysis of the EuroSki, these very large treatment discontinuation study from Europe that Francois Mahon presented, again showing the the good number of patients that can achieve this remission. Remember, in this study, the criteria was MR4 for at least 1 year, although the majority of patients had been in deeper molecular responses, meaning MR 4.5 and for longer than 1 year. So, you know, just confirming the value of this approach. A fascinating study on treatment-free remission in children. This came from our colleagues from India and Mumbai, where, you know, and this is very important because we don't have as much information in children. And of course, for children, stopping therapy has even a higher dimension, and showing the possibility of getting these very good results, a very large number of patients, and incredibly valuable, again, because it's the first experience that demonstrates these. And then a couple of studies talking about patients that are outside of our current recommendations, patients who don't really meet the criteria that we have established for treatment discontinuation, that they don't have quite as deep a response, that they don't have quite as much of a duration, and that was presented by Dr. Bonifacio from Verona. And then another study looking at patients that have had resistance to prior tyrosine kinase inhibitors, that was presented by Delphine Rea. And both of them show that, yes, some patients will have a sustained remission after treatment discontinuation. Of course, these probabilities are much lower. So it is not something that we recommend. I mean, it is important to understand that for some patients where there are circumstances that necessitate the interruption of therapy, in that case, well, there is a possibility. It, of course, needs greater surveillance and monitoring, but it is not the standard recommendation. But again, we recognize that in some instances that has to happen because of toxicities or other circumstances, and so this information then becomes valuable. Well, I could keep going on because there was a lot of very good data presented, but I don't want to eat into Daniela's time, because we also had fantastic presentations from our research studies and the basic science in CML. So I'm gonna close here. I'm happy to take any questions after Daniela's presentation, and I'm gonna turn it back to Stephanie.